Why do some plants grow better than others? One reason for this might be mycorrhizal fungi. What do these mycorrhizal fungi do for plants?
Hello, I am Adriana.
Hello, my name is Sophia.
And hello there. I'm Dennis, and welcome to our presentation.
Many of you may be wondering what a symbiotic relationship entails. It is quite simple really, as it is a relationship between two species that provides at least one of them benefits. There are three main types found in nature. These include mutualism, commensalism, and parasitism. Mutualism is the most beneficial as both participants in the relationship benefit. A great abstract example of this could be found when looking at our relationship with dogs. Dogs rely on us for their emotional and physical well-being, while we often rely on dogs for companionship or assistance in everyday tasks in the case of service dogs. Commensalism is similar to mutualism in that one participant benefits, but the second participant does not benefit, nor are they harmed. Parasitism, however, results in the benefit of one organism at the detriment of another. For example, leeches or other worms that have hosts in order to survive and receive their proper nutrients. In the case of our project today, we are looking at a mutualistic relationship between plants and mycorrhizal fungi.
So what are mycorrhizal associations? Mycorrhizae are symbiotic associations that form between the roots of most plant species and fungi. These symbioses are characterized by bidirectional movement of nutrients, where carbon flows to the fungus and inorganic nutrients move to the plant, thereby providing a critical linkage between plant root and soil. In infertile soils, nutrients taken up by the mycorrhizal fungi can lead to improved plant growth and reproduction. As a result, mycorrhizal plants are often more competitive and better able to tolerate environmental stresses than non-mycorrhizal plants. Plant responses to colonization by mycorrhizal fungi can range from dramatic growth promotion to growth depression. Factors affecting this response include mycorrhizal dependency of the host crop, the nutrient status of the soil, and the inoculum potential of the mycorrhizal. So when did these relationships evolve? The earliest land plants, 407 million years ago, were associated with fungi producing mycorrhizal-like intracellular structures similar to extant symbioses involving glomerulomycotina and mucoromycotina. Arbuscular mycorrhizal endosymbioses then diversified by the late Carboniferous period. Pinnaceae species from the late Jurassic and early Cretaceous formed the first ectomycorrhizal associations involving dikarya. Types of associations. There are four main types of associations. The first and the most widespread is arbuscular endomycorrhizae. This grows in between the cells of a plant root, but also forms arbuscules, which are structures that allow the fungus to penetrate the actual cells in the plant root. This type of mycorrhizae is more invasive than that of ectomycorrhizae. A diagnostic feature of arbuscular mycorrhizae is the development of highly branched arbuscule within the root cortical cells. The fungus initially grows between the cortical cells, but soon penetrates the host cell wall and grows within the cell. The second type is ectomycorrhizae. This produces a system of hyphae called the Hartig net in between the cells in the root of a plant. The mycorrhizae is within the plant root, but does not enter the actual cells of the roots and instead travels around the root cells. This fungus also forms a mantle around the outside of the roots of plants and increases the surface area of the roots and acts as a shield against pathogens. With this shield, the endomycorrhizae are able to increase the plant's ability to obtain nutrients and water. The third type is ericoid. The term ericaceous is applied to mycorrhizal associations found on plants in the order Achilles. The hyphae within the root can penetrate cortical cells, however, no arbuscules are formed. There are three major forms of Ericaceae mycorrhiza. And the fourth and final type is orchid. Mycorrhizal fungi have a unique role in the life cycle of plants, orchid plants. Orchids typically have very small seeds with a very little nutrient reserve. So these plants become colonized shortly after germination, and the mycorrhizal fungus supplies carbon and vitamins to the developing embryo. So what are some types of plants that have mycorrhizal associations? Well, the majority of vascular plants are mycorrhizal. 72% are abuscular mycorrhizal, 2% are ectomycorrhizal, 1.5% are ericoid, and 10% are orchid mycorrhizal. Just 8% are completely non-mycorrhizal, whereas 7% have inconsistent mycorrhizal associations. A conserved core module of the common symbiotic signaling pathway is shared by all host plants that establish endosymbioses, including arbuscular mycorrhizal and actinrhizal associations. This striking conservation among widely divergent host species underlies the shared evolutionary origin for this ancient symbiotic signaling pathway. A common feature of all these mutualistic associations is phytohormone associated modifications of the root development, which lead to an increase in potential colonization sites as well as major structural and functional changes to the root during the establishment of symbiotic tissues. So types of plants without associations include azaleas, beets, blueberries, broccoli, Brussels sprouts, kale, cabbage, carnations, cauliflower, collards, cranberry, huckleberry, mustard, protea, rhododendron, sedge, and spinach. Review of mycorrhizal research. One paper by Miller titled The Role of Mycorrhizal Fungi in Soil Conservation looked at soil disturbance such as tillage, which can dramatically affect the function of mycorrhizae in an agricultural system. Miller documented an interesting case where disturbance of no-till soil resulted in reduced mycorrhizal development and subsequently less absorption of phosphorus by seedlings of maize in the field. The researchers hypothesized that soil disturbance reduced the effectiveness of mycorrhizal symbiosis, and to confirm this, they conducted a series of growth chamber studies with non-disturbed and disturbed soil cores collected from long-term field plots. Disturbance reduced both mycorrhizal colonization and phosphorus absorption by maize and wheat roots, but did not reduce the phosphorus absorption by two non-mycorrhizal crops, spinach and canola. So the authors concluded that under nutrient-limited conditions, the ability of mycorrhizal seedlings to associate with intact hyphal networks in the soil may be highly advantageous for crop establishment. Another paper by Thompson titled Decline of Vesicular Arbuscular Mycorrhizae in Long Fallow Disorder of Field Crops looked at crop rotation and fallow systems, which can affect the diversity and function of mycorrhizal fungi. Thompson described the role of fungi in a long fallow, more than 12 months, disordered of field crops in a state of Queensland, Australia. In semi-arid cropping systems, cleaned fallows conserve soil moisture and nitrate for subsequent crops. Since the 1940s, some crops sown immediately after a long fallow grew poorly and had phosphorus and zinc deficiencies. Australian researchers found that the fallow resulted in a decline of propagules of fungi in the soil and reduced colonization of crop plants in the fields. And furthermore, they conducted inoculation trials and found that increasing inoculum abundance in the soil overcame the impact of fallow. They recommended that farmers avoid planting mycorrhizal dependent crops such as linseed, sunflower, and soybean following periods of fallow or after a non-host plant such as canola that led to reduction in AM propagules. One final paper entitled Inoculation of Pinus with Ectomycorrhizal Fungi in Puerto Rico by Vozo looks at pines that were not native to Puerto Rico, and their fungal symbionts were absent from the soil. As far back as the 1930s, there had been an attempt to establish pines on the island, but they were very unsuccessful. Typically, pines germinated well and grew to heights of 8 to 10 centimeters in a relatively short time, but then after that rapidly declined. The addition of phosphorus fertilizers did not substantially improve the plants either. So in 1955, soil from under pines in northern Carolina was transported to Puerto Rico, where it was incorporated as an inoculum into the soil around the 1-year-old plants. 32 seedlings were inoculated, and an equal number were monitored as non-inoculated control plants. Within 1 year, inoculated plants had an abundant mycorrhizal colonization and had achieved heights of up to 1.5 meters, while most of the non-inoculated plants had died. Further trials with mixtures of surface soil containing mycorrhizal fungi with pure inoculum, consisting of fungi growing in a peat-based medium, confirmed that inoculated seedlings were consistently more vigorous and larger than non-mycorrhizal ones. Subsequent surveys, more than 15 years after inoculation, indicated that inoculated fungi continue to grow in the pine plantations.
Now that we have talked about what are symbiotic relationships, how do these relationships benefit an organism? Some benefits include protection, better nutrient access, or even the ability to reproduce easier. In plants, these benefits can be increased survivability to an increase in nutrient and water uptake, as well as disease resistance. Symbiotic relationships not only help the user survive, this relationship was also seen to help the diversity of plants. As land plants evolved early on, root systems were integral to have in order to thrive and survive. Without roots, plants may not have been able to survive as well as they did. With the help of mycorrhizal associations, plants could get a good hold on the new world. Plant symbiotic relationships are mutualistic relationships and that both entities benefit in the exchange. This means that both survive and thrive with one another's help. Now, the benefits of having a mycorrhizal relationship with plants is that the fact that most fungi attach to the roots and add more to the root system. As we know, adding more surface area to roots helps to increase the uptake of water and nutrients, thus increasing the growth and survivability of the plant host that the fungus associates with. The fungus then benefits from the plant since the plant will provide carbohydrates necessary to the fungus within their cells. With this extra surface area and nutrient uptakes, plants can better survive an environment to be able to pass on their genes to the next generation. In conclusion, plants and mycorrhizal fungi have a very long history. Both benefit from one another and help each other survive and thrive within their environment. The fungus is already among us, and it is very much here to stay as it is an important part of our ecosystem, but also important to the crops that are grown in our world and everything that we do. Thank you all for joining us on this presentation about mycorrhizal fungi. We hope that you learned something new that you didn't know before. Have a wonderful day.